So thank you. I'm really excited to share today about some of the possible mechanisms that can underlie long COVID and how long COVID may have effect on Alzheimer's disease-like symptoms. So the COVID-19 pandemic has gave us a complex clinical landscape, and it doesn't end at the resolution of acute infection. So for many, they have symptoms that will linger post-virus, and these symptoms have been called long COVID, post-COVID complications, and post-acute sequelae of COVID-19. So these lingering symptoms can result in multi-organ damage, and this can give rise to symptoms such as fatigue, shortness of breath, and cognitive dysfunction. However, the central mechanisms that underlie these post-COVID-19 still remains unclear. So the COVID-19 virus can aggregate in brain regions such as the hippocampus, the brainstem, and cerebral blood vessels, and we all know these are regions that are associated with memory and cognition. So a mild respiratory SARS-CoV-2 infection has been shown to activate microglia, and this microglia activation results in increased inflammation, and this can then damage the synapses and the cytoskeleton, which can worsen neurodegenerative diseases, and it can reduce cognitive function. So COVID-19 has also been associated with elevation of plasma neurofilament light chain, reduced neurogenesis, an increase of pro-inflammatory markers, as well as excessive activation of our immune responses. So taken together, it's clear that COVID-19 plays a role in cognitive impairment. However, the viral hypothesis of which this occurs through still remains unknown. So one possible mechanism for this could be the kallikrein-kinin system. So the kallikrein-kinin system is a family of vasoactive peptides, and they have been implicated in inflammation and vascular complications of this virus, and could potentially play a role in the development of more severe symptoms, such as long-term cardiovascular damage, as well as acute respiratory distress. So within the kallikrein-kinin system, we see common kinins that are upregulated in COVID-19, and so one study has shown that in COVID-19, there's an increase of plasma kallikrein, high molecular weight kininogens, and bradykinin-like fragments. However, there's also a reduction in bradykinin itself, which signifies that dysfunction of this kallikrein-kinin system could play a role in COVID, and it could also play a role in some of the neuroinflammation we're seeing in long COVID. So another study has shown that in COVID-19 patients, they see an upregulation of B1R in the liver and lung. So B1R is one of the final products of this kallikrein-kinin system, and it has played a large role in inflammation in various studies, and to date, it has been shown to play a large role in stroke, hypertension, and heart failure. And since this acts primarily through inflammatory mechanisms, we can suggest that B1R possibly plays a role in some of the neuroinflammation seen in long COVID. So in order to really study the role of B1R in long COVID, we first needed to characterize a model of long COVID. And so to do this, we use K18-HACE2 mice, which these mice are genetically modified to express the human ACE2, which allows them to become infected with the virus. So these mice will receive a low dose of COVID-19, and this dose gives rise to about 80% survival in our mice, and then we can diagnose them with about a mild infection. So after 10 days post-infection, we don't see any significant changes in body weight. However, at 45 days post-infection, we see a significant increase in IgG antibodies in our infected group compared to our mock. And so this allows us to know that this virus was getting into these mice. So we found increase of the viral S spike protein in our 10 days post-infected mice, as well as an increase of double-stranded RNA, which indicates an active replicating virus. However, at 45 days in post-infection, we see that there's no longer a viral S protein or double-stranded RNA in our infected group, which signifies that this antibody production we saw earlier can occur independently of viral replication. So to look at some neurocognitive symptoms, we're able to show that infected mice who have long COVID display behavioral alterations such as elevated anxiety, attenuated exploratory behavior, as well as an altered short-term spatial memory. A hallmark of long COVID is lung fibrosis. And so we confirmed this in our model, and we were able to show an increase of collagen production following infection. And this was actually correlated with an increased B1R expression, as shown here with linear analysis. So this lets us know that B1R may be having a part in this long COVID infection. And so we then looked in the amygdala, hippocampus, and prefrontal cortex, and we were able to show that there was increased B1R expression in all these key regions in our infected group. So this work so far was published last year in Cells, but since then, many studies have shown that COVID-19 may be implicated in Alzheimer's disease-like symptoms, and they find that this microglia activation can cause this neuroinflammatory environment, causing this phosphorylation of tau, and eventually leading to an Alzheimer's-like disease. However, the role of B1R in SARS-CoV-2 induced exacerbation of Alzheimer's disease and the related dementia pathology still remain understudied. So another clinical study showed that there's hyperphosphorylation of tau in COVID-19 patients, and this was at serine 262 and 396, and they further confirmed this in neuronal cells, and they were able to show that tau is mislocalized and overspreading in the cytoplasm of these neurons. So this allows us to suggest that SARS-CoV-2 infection in neurons causes tau pathological changes, and this could contribute to some of the neuro-COVID symptoms that we see and why it may be impairing some of this neurohomeostasis. So because of these altered behavioral functions, this increase in B1R expression, and an increase in tau phosphorylation, we hypothesize that B1R blockade will reduce the persistent neuroinflammation and tau phosphorylation that we see in SARS infection. So to do this, we use the same model we used before. However, this time, on the same day of infection, they received osmotic mini pumps with SSR 240612. And so this is a B1R specific antagonist, so it really inhibits that B1R, and it can cross the blood-brain barrier, allowing us to look at this neuroinflammation and cognitive symptoms that are associated. And so they're given this for 28 days, and following 28 days, we continue our experiments where we look for microglia morphology, microglia activation states, as well as tau phosphorylation. So reactive microgliosis plays a large role in inflammation, as a neurotoxic trigger such as inflammation or infection can damage these neurons and cause them to secrete factors that can activate microglia. In turn, these activated microglia secrete pro-inflammatory factors that further damage the neuron, and it creates this perpetual cycle of neuroinflammation, and this is often seen in Alzheimer's. So to kind of study this in our model, we stained our mouse brains in the amygdala for IBA1, which is a microglia stain, and we really want to look at the microglia morphology. And as you can see here, our SARS group has more activated microglia as indicated by an increased soma size and a retraction of their processes. However, when we block B1R in our infected group, we see a reduction of this and an increase in processes relative to our mock control. So we did quantify this using the skeletonized ImageJ plugin, and from this we were just able to confirm that the soma size was increased in SARS infection, and that their microglia processes are being retracted by a decrease in branch length, junction, and voxels, and that B1R blockade is able to reverse this, signifying that COVID-19 induced microglia activation may in part be mediated by this B1 receptor. So to take it a step further from just activation, we wanted to look at this activation is pro-inflammatory or anti-inflammatory. So in microglia, they can either be a pro-inflammatory phenotype, where this simulates neurotoxicity and creates this really neurotoxic and neuroinflammation environment that can further damage neurons and worsens Alzheimer's disease. However, you can also have anti-inflammatory microglia that try to help protect the environment and further continue our healthy neurons to try to combat this. So we use CD86 to mimic our inflammatory microglia and CD206 for our anti-inflammatory, and we found that in our infected group, there's an increase of CD86 positive microglia, whereas there's a reduction in CD206 positive microglia compared to our mock-treated group. However, when we block with B1R, we see that this shifts it to a more anti-inflammatory phenotype following infection, once again thinking that maybe B1R can block this inflammatory microglia activation. So we quantified this and also wanted to further confirm this, so therefore we measured levels of TNF-alpha and IL-6, as these can also signs of inflammation with microglia, and we found an increase of TNF and IL-6 positive microglia cells in our infected treated group, and once again B1R blockade was able to reverse this. So to kind of tie this all together, we know that inflammatory stimuli can result in microglia activation and subsequent neuroinflammation, and this has been shown previously to cause tau phosphorylation, and this hyperphosphorylation of tau leads to a disintegration of these tubules and the increase of neurofibrillary tangles, which can result in neuronal death and subsequent Alzheimer's disease. So in our model, we measured phospho-tau, and so this is 8T8, and we see that our infected group has a significant upregulation of tau expression. However, when we block B1R, we are able to see a reduction of this, which once again verifies that B1R is somehow mediating the COVID-19's induced Alzheimer's-like effects. And so from this study so far, we can conclude that microglia activation is induced by COVID-19 infection and that it takes on a more pro-inflammatory phenotype. However, when we block B1R, we're able to shift this balance to more of an anti-inflammatory environment, so it may be able to combat some of these effects we're seeing in long COVID patients. We also found that a single acute infection of SARS-CoV-2 was able to increase tau phosphorylation, whereas when we block B1R, we're able to reduce this partially, which allows us to believe that hopefully B1R antagonism may eventually be a promising therapeutic in order to prevent or reduce some of these Alzheimer's disease-like symptoms that occur following COVID-19 infection. And I would like to thank all our collaborators and our funding sources. Thank you.
Curious, just quickly, how difficult were these experiments to do based on the fact, were you required to do most of what you showed in BSL-3? Or once you paraffinize it, are you okay for all of your stains to be downgraded to BSL-2?
So we work in a collaboration. So Dr. Eels and Hannah over here actually do all the BSL-3 work. And then once they're in paraffin, they give them to us, and we'll do all the immunohistology work. So it's definitely a tag team process. It's a very intricate process to get them out of BSL-3, so It definitely takes a team.
Great. And Elizabeth, do you have a question? Yeah, we have time for one more.
Beautiful work, Duke. Did I understand correctly that you analyzed microglia in the amygdala? Mm-hmm. So why amygdala?
So in previous study, we showed that the amygdala and hippocampus were playing a role with some of our behavioral changes we saw. And we have the data as well for the hippocampus, but it was not analyzed in time. But yeah, so we did it based off our previous studies.
Thank you.
Mm-hmm.